This is your fall break assignment, and we're still working on organic chemistry. Today we're going to talk about isomers, which essentially means compounds that have the same molecular formulas but different chemical and physical properties. You notice there's lots of different ways to write isomers when we were naming compounds, and the name specifies exactly which isomer we're working with if you have a structural isomer. All of these compounds that are shown could have a same molecular formula, but their atoms are connected in different ways. For example, these are all 6 carbons, I think. 1, 2, 3, 4, 5, 6. Yep, there's a 6th carbon, but we wouldn't call this hexane because this 6th carbon is hanging off of another carbon in a 5-carbon chain. So we would call this one 2-methylpentane. So you already learned how to name these. I'm not going to name all of them. I just want to show the difference here. In this one I have 1, 2, 3, 4, 5, 6 carbons. But these carbons, number 5 and 6, are hanging off of a 4-carbon chain. So this would be called 2-ethylbutane. How about this guy over here? 1, 2, 3, or 1, 2, 3, 4. So we'll have to go with the 4-carbon chain for butane, right? But on the butane, I have 2 methyls. So I call this 2,2-dimethylbutane. How about this guy? 1, 2, 3, 4, 5, 6 carbons. But since I'm bonded to all other carbons, I have 2 hydrogens, 2, 2, 2, 2, 2. So this won't have exactly the same formula. I have 6 carbons, but for every carbon, only 2 hydrogens, not the normal 2n plus 2 that we saw with the alkanes. So structural or isomers are the ones that have the same molecular formulas, but their atoms are connected in different ways. This arrangement of atom creates a different chemical property and a different physical property, such as density or boiling point or whatever. So if you had these formulas for heptane, C7H16, then the 9 different isomers would have different names and formulas for each one. So we'll write the structural formula and the name. Boy, that's a lot of work. Here we go. We better get to it. So this is just normal heptane, N-heptane. You could just call it heptane, and everybody would understand that you simply mean a 7-carbon chain in a straight row. Now if we have 7 carbons where 2 of them are hanging off, then we're going to number this in such a way that we get the lowest numbering. So no matter if I start from left to right or right to left, I'm going to get the same numbers either way. So we'll call this the 2,4-dimethylpentane. And remember, anytime you have a number and a letter, you put a hyphen in between. So I need 2,4-dimethyl, right? And we put commas between numbers. That's also in your packet. In case you forget about the hierarchy of which rules to apply in which order, I gave you a cheat sheet in your packet. Now let's look at this one. 1, 2, 3, 4 carbons this way, or 3. Yeah, looks like 4 is the highest number we can get for a chain. So we'll call this 2,2-dimethyl. And then what? Nope, 2,2,3-trimethyl, huh? 2,2,3-trimethyl. And then that is butane, my 4-carbon chain. 2,2,3-trimethylbutane. There we go. That's it. How about part F? 1, 2, 3, 4, 5, 6. And my 3rd carbon is my lowest number I can get. So 3-methylhexane. One, 2, 3, 4, 5-carbon chain. So 2,2-dimethylpentane. Hey, didn't we have that? No, we had a 2,4-dimethylpentane. Dimethylpentane. There we go. All right, how about 2-methylhexane? What does that look like? So we draw the hexane, 4, 5, 6, and off the second carbon, put a methyl group there, right? And then 3-ethylpentane means we've got to have 5 carbons in a row, and hanging off of my third one, which is my middle one, my ethyl group, C2H5. You could also show it as CH2, then CH3. That's allowed. You know, an ethyl group, I don't see one hanging off of here. Ethyl groups we could write as CH2, CH3. That's the same as C2H5. So that's how we deal with naming these. They're called isomers because they have the same empirical formula, right? The same multimole ratio of carbons to hydrogens, but they're organized in a different way, so they are called isomers. Stereoisomers are a bit more complicated because the compounds have the same connections between atoms. So the atoms are connected to the exact same other atoms in the molecule, but we have different arrangements of the atoms in space. So, stereoisomers, bear with me. Let's have a look at these guys. Suppose that we have a cyclopentane. Here's my cyclopentane. Now what I'm going to do with this for you, I've shown the dash wedge structure so that you can see what's coming towards you and what's coming away from you, but I'd also like you to see what it looks like if I were to tilt this on its y-axis so that you can see the side view. So again, the dash wedge structure, this wedge means this is coming towards you, all right, and this is the side that is facing you. So coming towards you off of this compound is a bromine and a bromine. They're on the same side of this cycle here, on the same side of the carbon. And then if I were to flip it, then I would have a bromine on top right next to a hydrogen. See how this crowds our bromines? This is a less stable configuration for the bromopentane than if we were to put them across from one another, so that now the bromine's next to a smaller hydrogen and this bromine's next to a smaller hydrogen. This is much more stable than the other configuration of atoms. So this more stable compound, we call it trans, to indicate that they're across from one another. This is one type of stereoisomer we could have, is the cis-trans configuration. Cis would be on the same side, and trans would mean they're across. The same side is more crowded, so that tends to be less stable. This is a pretty critical type of configuration. Let me clear the writing. There we go. Especially in your body. You've heard of trans fats. Well, that word has meaning. Trans means they're arranged across from one another. So the cis-trans configuration in an isomer happens in your eye so that you can perceive light. As a light wave hits the retinol, the cis-retinal in your eye, derived from vitamin A. Vitamin A is retinol. OL means it's an alcohol group. And then your body takes that vitamin and turns it into retinal, which means it's now an aldehyde. So we've transferred the OH into a double bonded O, right, to make an aldehyde out of it. So what happens when the light strikes it is that we take this particular bond where we have ligands on the same side and we flip them across from one another. So now they are a trans, and it changes the shape of the molecule so that on the retina of your eye, the light stimulates a change in the shape of the molecule, which sends a signal to your brain, and now you see something. Your brain gets the signal, hey, you're looking at something. We interpret the photons of light as colors and shapes that we know the names of and we can recognize. So these molecular shapes tend to be critical in your body. Your body processes things differently according to their different shapes. So let's again look at that cis-trans isomer. Cis means they're on the same side of the double bond. Trans means they're arranged on opposite sides of a double bond. So or on the cycle, in the cycloalkane. So if I put my chlorines here, and hydrogens then here and here, then they're on the same side, right? That would be the cis configuration of dichloroethene. So now let's put my chlorines across from one another. This would give each chlorine a lot more wiggle room for its electrons to take up a lot of space, right? Hydrogen doesn't take up hardly any space. When they're arranged across from one another, then this is called the trans configuration, whereas this one was the cis. That's how we can control what types of things we get in pharmacy or biochemistry or whatever we want to do. There's another type of stereoisomer that has to do with mirror images. We call them right-handed and left-handed enantiomers. In other words, optical isomers. That's what an enantiomer is, an optical isomer, according to which direction they rotate plane polarized light. Not plane as in not very exciting, but plane as in light that's traveling along a plane, a three-dimensional plane, right? Louis Pasteur was not the first guy to identify isomerism this way, but he was the first one to actually be able to sort out the crystals of a compound, tartaric acid, that would turn the light to the left or to the right, because the crystals looked different. They're visually different, so he was able to separate them using forceps and a hand lens. Sounds pretty tedious. It would be like trying to separate salt crystals from sugar crystals using tweezers and a magnifying glass. But basically, when you put the enantiomers in solution, they turn the light either away from the perpendicular or towards the perpendicular. Can't even see that color. Let's try this. So here's the plane that my light's going at. So if we're turning it away or turning it towards, then we get a different handedness, they call it. Dextrorotatory isomers rotates the plane to the right, where dexterity and dextro, or the prefix D, means right-handed. And then levorotatory isomers rotates the plane to the left. So this is our L for left. They also used to call this the S configuration for the Latin sinistra, which is left-handed, as opposed to dextro, which was right-handed. You know how they used to do in biology, everything named for the Latin terms. It's a good thing they've started naming things in English now, right? That's the way I see it anyway. What's interesting, though, about stereoisomers is that living organisms tend to use only one isomer. In your sugars, it tends to be the right-handed configuration that's preferred. So if I were to feed somebody L-glucose, the left-handed type of sugar, they would simply starve to death because your cells don't know what to do with this. They don't recognize it, they can't process it, and they wouldn't survive. So it's the right-handed glucose that your cells need. In amino acids, strangely enough, the amino acids are preferentially left-handed. So the L configuration is produced by living things and absorbed by living things. They cannot absorb the wrong-handed types of molecules. Weird, I know, but that's the way it is. So that's what isomerism is. There's a lot more to it than that. These are the ones you need to know, that structural isomers have the same atoms just arranged in different places. But a stereoisomer has all of the atoms bonded to the same things, but they're geometrically in a different place in three-dimensional space. That's it for isomers. I'll see you guys soon.